Hello. We would have just recently concluded our lecture on plant cell types and tissue types. Consider that this is our first lecture and your first introduction to our plant physiology. I thought it would be prudent to make a summary video of what we looked at. I know you are all thinking that plants are so boring, but I'm just going to try to help you to ease yourselves into the course. So quickly, let's look at a summary of what we looked at this week in class. Okay. All right. Okay, so one of the first cell types that we looked at in class would have been the parenchymal cells, right? And we started with that one for no more reason than the fact that it is the less complicated, right, of the three types that we spoke about. Now, what is unique about it? How can you identify whether or not you're looking at a parenchymal cell? Parenchyma cell is going to be thin in nature. So its primary walls are going to be very thin. As you can see in this image here, looking at these walls, it is going to be that cell which functions in undifferentiated growth. So while you can see the nucleus, you're not really seeing a lot more happening there. Parenchyma cells are responsible for functioning in storage of starch, for instance. So when we think about the roots and tubers that we love to eat, like Irish potato or cassava and sweet potato, it is because of the parenchyma cells that are storing starch that gives those roots and tubers their ability to bulk up. So parenchyma is also responsible for functioning in photosynthesis as well. Now, these cells are going to be alive once they become mature. Then another cell that we spoke about is the collenchyma cells. And if we are to look in this image here, what do we notice about the collenchyma? We're seeing that the cell walls are looking a little bit more thickened, right? And usually the collenchyma cells are going to be found just immediately after the dermal layer or the epidermis. The collenchyma cells, what is going to be unique about them? What is causing that thickening in their cell wall? It is going to be because they have accumulated pectin as well as cellulose. And now that they have accumulated pectin as well as cellulose, I mean, parenchyma has cellulose, but not pectin as well. That's now telling us that, these cells are going to be a little bit more rigid, but they are going to be a bit plastic so they can stretch, they can allow for some amount of flexibility wherever they're present, right? So you may find them in leaves, in petioles, and in stems as well. Oh, moving on to the hardier guys, the sclerenchyma. When we think about the sclerenchyma, look at these cells. You're seeing that you have ridges present, and you're seeing that the cell wall looks a little bit more advanced, a little bit more developed. This is saying to us that there is more thickening that is happening. So here we're actually getting secondary cell walls coming into play that have been deposited with lignin. And when we think about lignin, we need to think about rigidity. We need to think about hardiness. So think about the woody texture of a trunk of a tree. All right, think about that. Sclerenchyma is what is usually going to be deposited there. And because of how thick it is going to be, then you tend to see that, okay, it's going to be dead at maturity. So parenchymal cell will also have suberin and cutin in their walls. And this tells us that there's going to be some amount of waterproofing that is going to come into play. Here's a table that just looks at the structure as well as the function of the plant's cells side by side, right? So as we move from the parenchyma cells all the way up to the parenchyma, what do we think about? Think about thickening in cell walls. Think about cell wall composition increasing. So you think about pectin being added and to parenchyma, think about lignin, suberin, as well as cutin being added to sclerenchyma. And then think about the plant being less flexible wherever those cells are going to be deposited as you move from the parenchyma to the sclerenchyma cells. And remember, when I say move, I'm just thinking your thought, how you think about cells based on their structural features. Now, we need to then have tissues. So cells then help us to build tissues, right? So one of the tissue types that is of high relevance in the plant is the vascular tissue. Now, the vascular tissue is made up of what we call the vascular bundle, which is made up of the xylem and the phloem. Look here. So the xylem is going to be made up of tracheids as well as vessel elements. And what are going to be their roles? Their role is to transport water as well as mineral elements or nutrients from the soil. Now, if it's taking it up from the soil, that means that the movement through the xylem is going to be from soil upwards. So there's one direction that this water as well as the nutrients which are dissolved in the water or in solution are going to move. Whereas in the phloem, which is made up of the sieve elements, made up of companion cells, right? They are going to have sugar or the photosynthase, which are produced where? In the leaves of the plants, move through that tissue. Now, unlike water, which moves unidirectionally, phloem will have its sugar transport moving in several directions. The sugars will move from the leaves to the stem to the roots. Sugars may move from the leaves to a fruit to a developing bud, etc. All right. Now, it's important to note that in a dicot stem, and what is a dicot stem again? A eudicot plant or a dicot stem is going to be on a plant that has two cotyledons, for instance. And if you were to look on the leaves, then you'd see the venation looking reticulate or netted. If you were to look at a monocot stem, then you'd see that the leaves will have their venation moving parallel. And an example of a monocot plant is corn. An example of a dicot plant is a bean, for instance. So if you were to cut cross section this way, right, of both a dicot stem and a monocot stem, what would you see? The vascular bundles would be arranged in a circle in the dicot, whereas in a monocot, they would be scattered all around, right? There's no true format for their arrangement. And then in these regions is where you find ground tissue. So what is ground tissue? Collenchyma, parenchyma, those make up the ground tissues. And then dermal tissue, another major tissue that is found in plants. So when we think about dermal, we think about dermatology. And when we think about dermatology, we think about the skin, right? So we're thinking about the upper surface of the plant or the surface, the outer surface of the plant. So that surface is going to be made up of cuticle or cutin, which basically helps to prevent desiccation or drying in the plant because of how hot it is outside or how dry it is, life on land is. Then below that, you'll have the upper epidermis, right? So you find the dermal tissues. Now, we are going to have growth occurring in the plant. And where we have elongation in the plant is where we have growth occurring at the apical region as well as the root tips, right? So that is primary growth. We also have secondary growth occurring, which will allow for expansion in girth, right, of the plant. Now, it is these cells that we spoke about, parenchyma, florenchyma, sclerenchyma, which are going to be at those sites of division, right? So at the meristematic region is where you're going to have these high energy activities occurring where the cells are going to be rapidly dividing, multiplying through mitosis. Now, in a plant that is not woody, right? Like our monocots, you will tend to not find secondary growth occurring. You're not going to find the thickening of the stem, for instance, right? You'll only find like a primary structure will occur because of the vascular bundles that are there. But you'll find secondary xylem, secondary phloem occurring in your eudicote plants and in those stems, which gives that expansion. All right. I trust that this summary was good for you, and I'll see you in class. Take care.